Hey guys, this is JB Mom from Planning Seeds Tutoring. In today's video, we will check out the central dogma of molecular biology, or in other words, just the central dogma of bio. Don't forget to use my referral code JBMON20 to get 10% off on your next tutoring package. The central dogma is a term coined by the British molecular biologist Francis Crick in 1958. Probably known from Watson and Crick, which is who discovered the double helical structure of the DNA. And the dogma itself, the word itself, the term itself just refers to the known methods of genetic information transfer between nucleic acids and proteins. And it pretty much just states that in general, DNA can be copied into itself via replication. It can be copied into being mRNA via transcription. And mRNA specifically, which is a type of RNA. And RNA can be coders for proteins via translation. And just to reemphasize, don't forget that DNA is a double-stranded structure, has its helical, and it has the four nucleotides: adenine, thymine, guanine, and cytosine. Adenine and thymine are base pairs, and guanine and cytosine are base pairs. And in RNA, instead of having thymine, it has uracil. All right. So we know that there are trillions of cells in the human body, and almost all of them have the same DNA in them. Notable exceptions, though, are gametes, which are the reproductive cells, amongst a few other types of cells. And for all these cells to have the same exact DNA, there must be some kind of mechanism to copy the DNA, because cell theory pretty much states that all cells come from pre-existing cells, which implies kind of that the DNA also has to get copied from that very first cell. This process is replication. So replication overall just comes down to a few steps and a number of enzymes, at least the basic understanding of it. Helicase, which is this enzyme right here. Helicase pretty much slices through the hydrogen bonds within the DNA to separate the two strands. Remember, DNA is made out of two strands, and it's a helical structure. And then we have topoisomerase, which is used to ensure that the DNA does not supercoil. Else, the DNA itself would end up bound together, and the strands would not be separated enough to continue with the replication. The DNA polymerase, pretty much, which comes in later, would not be able to go through. Primase is primase, which is an enzyme that I did not show here, it attaches this primer here, an RNA primer here, which these are RNA primers. And these RNA primers, they signal where the DNA polymerase, which is going to be this DNA polymerase, where the DNA polymerase is supposed to start attaching complementary nucleotides to each strand. So the DNA polymerase is pretty much what creates the new strand itself. So DNA polymerase, though, it can only synthesize new strands in one direction, the 5' to 3' end. But these strands, there are two strands inside of the for DNA, and they're going anti-parallel to one another, which means that they have both directions, 3' to 5' and 5' to 3'. So this is kind of like being a car that's only able to go in one direction, but it needs to drop off people in both sides of the two-way road. So this means that DNA polymerase actually starts at the 3' end on the strand that it's copying. And it attaches primers in the 5' to 3' direction. Not primers, attaches nucleotides in the 5' to 3' direction. Because the helicase itself is starting at it keeps unwinding the DNA. This means that in one strand, DNA polymerase keeps attaching nucleotides continuously. And that's what's happening in this strand itself. In this strand, the leading strand, it keeps attaching it continuously. But in the other strand, that's not exactly what happens. When helicase was about right here, we would have started the DNA polymerase over here. Well, as it goes a little bit further, we would have had a new RNA primer, which signals DNA polymerase to go that way. And then now we would have even more to go this way. Further on, we're attaching more and more strands in this direction. And this is what we call the lagging strand. In other words, the DNA polymerase has to keep restarting to attach the nucleotides because the helicase just keeps unwinding. Now, these little fragments right here, these are what we call Okazaki fragments. And these Okazaki fragments, as you can see, there's like a little gap between them. These gaps need to be mended by something called DNA ligase. DNA ligase. So overall, what we're going to see with replication is that this one strand of DNA, a double-stranded DNA, is going to lead us with two molecules of DNA. Well, both of these molecules of DNA are identical, and they're also identical to the original parent DNA itself. And then we also had to say that these copies are semi-conservative because each of these new DNA molecules, they actually have one strand from the parent, so what these two blues are, and the others two, which are the whites, get made by the DNA polymerase. All right. So now we have a lot of DNA because of replication. But how do we get this DNA to being the proteins that we need for our everyday functions? Well, we first actually need our DNA to be transferred into or copied into being messenger RNA, which is what we call mRNA. And this process is called transcription. And in transcription, RNA polymerase, which is different enzyme, very similar to DNA polymerase, it attaches nucleotides to make a complementary RNA strand to the template strand of DNA. This is going to be what we call the template strand, and the other is going to be the coding strand because it looks exactly like the RNA itself, except instead of having uracil, it has thymine. All right, so this yields us with just a standard mRNA. this mRNA now leaves the nucleus and it reaches the ribosome. Now, the ribosome is where the last general step of the central dogma still takes place, and that's translation. In translation, tRNA, which is another type of RNA, it carries an anticodon triplet. So an anticodon triplet, a codon is just a set of 3 different nucleotides. And the mRNA has what we call the triplet of RNA. The anticodons are going to be the nucleotides which match up to the codon itself. So in this case, we're going to have a uracil, which attaches to the adenine, cytosine, which attaches to the guanine, and cytosine, which attaches to guanine. And that's all part of the tRNA itself. So the tRNAs carry both these different kinds of anticodons, and they also carry separate amino acids based on these anticodons as well. Now, what happens is as they meet up with different section on the mRNA, the different codons in the mRNA, different tRNAs carrying different amino acids keep getting attached, which leads the amino acids to break away from the tRNA, and that's what yields us with a amino acid chain, which is exactly what a protein is. And that protein carries its own function, and that's the entirety of like the DNA to protein steps. There are a total of 20 amino acids, and each of these 20 different amino acids can be signaled by different codons in the mRNA, and that can be shown by a codon chart. So overall, all that the central dogma says is that DNA makes RNA, which makes proteins. Thank you all for watching, and I hope you guys enjoyed the video.